And you should be okay. So next we have Patrick Gorski. Patrick is a research scientist at the Wisconsin Department of Natural Resources. It's all yours.
Okay, thank you. Yeah, as just mentioned, so I'm a emerging contaminant research scientist at the DNR, but specifically I'm the Bureau of Water Quality. That means that one of the main things we look at in water quality is surface water. So lakes and rivers are the main thing that we're analyzing for PFAS and that we're monitoring for PFAS. So this is an outgrowth of PFAS surface water monitoring. Being that, what I did is we get some monitoring initiative funds from Region 5 EPA to specifically fine tune or look at how to improve or questions related to monitoring. So in this case, one of the questions that we have for surface water monitoring is for PFAS, are we collecting the correct surface water samples for monitoring? We in Wisconsin, we basically really dove into surface water monitoring in 2019, and the way that we've been monitoring it is collecting surface water grabs 6 inches below the surface. Okay, just ignore her. And so basically, if you don't know, so you just grab the bottle, you have the cap on, go 6 inches below the water, you open it, you fill it up, you close it, and you take it to the lab. So is that representative of the water body as a whole? And one question I really haven't seen answered is how does PFAS vary with depth in a water column, especially in a lake? My background is limnology, and as we know, certain things happen in lakes as they stratify in the summer. You have decomposition, you have a buildup of total phosphorus, you have, redox going on there, manganese, iron. My background is mercury research. You also have in certain lakes methylation of mercury. So there's a lot of, interesting dynamics going on, and I don't know how that relates to PFAS, and no one's really looked at that, and I really haven't. So that's a question I often get is like, I mean, we're trying to get statewide measurements, and we have over 200 samples, different sites, I should say, on the state, but we're, our first gut reaction was to collect that, the grab sample. Okay. Oh, okay. And then also, are we missing anything by analyzing for total PFAS? And when I say total PFAS, I'm not saying, I'm not talking about total organic fluorine or something like that has come up in a few of the other talks. But what I'm saying here is that you grab the sample, in that total, the whole sample is analyzed for PFAS. So that includes, any particulates that dissolve, particulate PFAS, that could be algae, that could be organic, inorganic particles, that could be the algae zooplankton, that could be microplastics with the microfilm and the salmonella attached to it or whatever, that we learned about in some of these other talks. So I worked with the lab to differentiate between dissolved and particulate fractions, because as we also know, that's important as far as transport questions, recycling, bioaccumulation, bioavailability, things, like I said, my background in mercury, that's important. Also, like for total phosphorus, what's available to plants and nutrients, and also transport down the system. If it's settling out, resuspending, okay, you get the idea. Lake Monona, why Lake Monona? Lake Monona is located right next to Madison. There's Mendota and Monona, so it's right, basically downtown. If you didn't know, Starkweather Creek, which flows into Lake Monona, has one of the highest PFAS concentrations in the state. It averages over 200 nanograms per liter for just PFOS. That drains from the Madison Airport. On one side of the airport, it's Truax Field, which is Air Force Fighter Wing. And there was widespread use of AFFF there, and it's been, basically constantly flowing into Lake Monona. So that's right out a back door, and it consistently has about 8 to 10 nanograms per liter of just PFOS in it, in the surface water. So it's an impacted site, and we now have a surface water standard in Wisconsin of 8. For PFOS, so it's above often our surface water standard. And then I also have a background, I did a lot of analysis. So as an analytical chemist, that's kind of nice to work with. In the statewide sampling that we've done, we actually, like, for the bulk of statewide sampling that we've done, often we'll get detects. As you probably know, if you analyze it, you're probably going to detect it. But the background concentrations or baseline are often really low in a lot of the rivers and streams. So if you're going to do a study and you want to like dig down into the sample analysis, it's nice to not have a bunch of non-detects and be able to have something to work with. So that's one of the scenarios I looked at or thought of as well. There's a fish consumption advisory on the lake, all the way downstream on the Yahara chain and the other lakes as well. So it's a health-related issue. And it's, like I said, a convenient location. It's actually, basically downtown Madison. We have the central office. We have a district there in the state lab of hygiene that we're working with. It's not one of those sites that you have to travel 4 hours away and it's and worry about transportation. So how I decided to take the vertical water sampling was to use a GoFlow bottle. And here's a picture of one. If you haven't seen one, that's actually us in the field. And you can see the state capitol over the top of the bottle there. So it's manufactured by General Oceanics. And I started using, or I have used one of these in the past when I was doing trace metal work. For trace metals, you can actually buy one with Teflon lined, but you don't want to use that for PFAS. So you just basically buy the basic PVC bottle. Ours is 5 liter. It's come in multiple sizes. The nice thing is that the sample doesn't touch a lot of anything besides the PVC. There's a nice ball valve at the top and bottom. And also it remains closed. So there's a few people that mentioned, foam and surface microlayers. So it remains closed, and you can go through that. And then the one that I had, there's a pressure release valve about 2/3 of the way down. And when it gets to depth, you open it up and then you send down the messenger and it closes it. So you capture it, you're not going through that surface microlayer, the sample container. And all the mechanisms are external. Like I said, it doesn't touch anything. You have all the gears outside, so the sample's not touching. Some things like a Niskin bottle, Van Doren or Kemmer bottle, they have all the stuff on the inside. And you can clean it. I talked to or contacted, the only other paper I found was this McInnes paper. And I worked off a protocol that they use and talked to a couple other researchers. So basically Alkanox, methanol, Millicue, didn't use the acid because we're not, some places use those for mercury. And the lab blanks were all non-detect. I worked on developing the particle and dissolved sample method with Chen Zhang at the State Lab of Hygiene. He has a whole bunch of method validation that I'm not going to cover right here because I want to get to the field results with you, but he fully vetted this with, blanks and spikes and uses standard reference material that he filtered and so forth. But basically what I did is took 2 samples. The first sample was for the PFAS analysis, and we filtered using a GFF filter, 1.5 micron. And the GFF filter was the best filter to show that, that it didn't contribute or suck up or anyway mess with PFAS. Anyway, so obviously you have the dissolved sample, and then you have the actual filter paper with the solid that you methanol extract to analyze there. So you have the liquid and the solid sample, and then not to pester the organics lab with getting a mass on that because I want to get nanograms per gram. I had the lab do their regular total suspended TSS analysis at the lab, but also had them ash that, put it into a 550 oven so it volatilizes the organics so I can get inorganic organic fraction. Anyway, so you have the various nanograms per liter sample and then the mass, and then you can get the nanograms per gram. Sorry to go through that really quick. But so I sampled, here are the results for August of last year. So this would be the worst case scenario for sampling. This is when Lake Monona, like many of the Madison lakes, is eutrophic, meaning that it's very productive. There's a lot of algae. So as you may or may not know, during the summer, lakes stratify. So the warm water is on top, and then you have the cold water on the bottom, and then you have the thermocline. And then also in a productive lake, you have the decomposition of the algae, and it's using up the oxygen. So you can see that. So starting with the left profile, that's the temperature, and you can see it's well stratified. And then also the red is the oxygen. So you have the well stratified lake. It was totally anoxic after 9 meters. Oh, I should also say, if you're unfamiliar with these graphs, on the top of the graph on the y-axis, that is, imagine the 0 is the surface of the lake and you're going deeper into the lake. If you haven't seen one of these before, and then the concentrations of the parameters are on the bottom on the x-axis. So as you go deeper in the lake, once you get to 9 meters, all the oxygen is gone. It's completely anoxic. And then so you have redox conditions going down there. When we're pulling up the sample, you could smell sulfur. I don't have that on here, but it was, so this is the worst case scenario. But so temperature, oxygen stratified. You also can see the green, the chlorophyll. There was an algal bloom. Also, not surprisingly, there was more TSS in the yellow. But when you look at the gray and the black, the gray is the PFOA, and then the black is the PFOS. It's basically the same all the way down to the top and bottom. So LG. So the PFAS was not stratified. It's basically the same top to bottom. I don't know if I was expecting that or not, but at least I measured it and can see that it's not stratified and it's within plus or minus 2 nanograms per liter. So this is the typical total PFOS measurements that I took. And yeah, so I want to break that down into total and dissolved. So again, the gray is the PFOA, the black is the PFAS. Blue is PFOA, and the reddish colors are PFAS. So starting from left to right, so the dark blue is PFOA in the particulate, and there was none. There was no PFOA, that was basically 0. It was all below detection limit. In the light blue is the dissolved PFOA, and as you can see, most of it, there's kind of a jump there at the top, but it's basically tracks. It's the same concentration as the total. But then if you look at the dark and the light red is the dissolved PFOA. And as you can see, it less of the total is dissolved. And then there's a sharp increase in the particulate concentration in the PFOS in nanograms per gram, culminating at the top of the bottom of the thermocline, the top of the hypolimnion, and then all of a sudden it goes away. And the reason that it goes away is that the TSS was non-detect. Unfortunately, there were no particles in the bottom. So the average, if you look down here, this is in dry weight, is 383 nanograms per gram dry weight. That's hard. I wanted to put that on some sort of like reference. If you assume that's 90% water, that'd be 38.3 nanograms per gram wet weight. And I did that because fish are measured in wet weight. In Wisconsin, fish advisories are at 10, 50, and 200. So 38 is above 10. So if you ate 8 ounces of that, you could only eat 8 ounces once per week. So there would be an advisory, but you could still eat it. Anyway, what are the other parameters associated with PFAS and the particles? The green is the algae again. As you can see, the algae decreases right above the hypolimnion. I've added the purple for the organic TSS in with the total TSS. And so the chlorophyll A and mass are similar. The mass was consistently 70 to 30% organic to inorganic. But as you can see, those don't have a similar trend as the PFOS, which increases right above the hypolimnion. The only thing that I could find that tracked with that was actually alkalinity. These are fairly hard lakes. So that'd be a measurement of the bicarbonate or the carbonate. So alkalinity seemed to trend similar with PFOS as the particles. And then at the 10-meter mark, there were fewer particles, but they're very concentrated. So I'm not sure what's going on or how like the sulfate functional group would be binding to another negative group. Actually, before I gave this talk, I gave a similar talk about this, but a graduate student in the same session was showing that, anyway, 1 minute. Okay, so this is a distribution of the blues are the carboxylics, the reds and oranges are the sulfonates. And as you can see, at the top, actually I have surface microlayer, the 6-inch grab, and then one down to 20. The 6 meter was duplicated. Anyway, it's pretty much consistent for all the different compounds percentage wise. And I feel confident about showing it percentage of total because the total PFAS was 30 to 35. So it's pretty much the same concentration. Those are the top compounds. PFOS 27%, PFOA was only 8%. Anyway, there was 6.2 at the bottom. But if you look at the dissolved is pretty much the same as the PFAS. I could put those side by side and pretty much the same. And if you look at the distribution of all the different compounds that were in the particles, they were all PFOS. So PFOS was the only particle that was bound to the particulates, top to bottom. And if you look at percentage wise, most of the PFOS was dissolved. And then a small amount in the particulates. It's usually 80 to 20 dissolved to particulate, consistent with depth, highly concentrated at right above there. So in conclusion, taking a 6-inch grab, that seems to be a good, we kind of lucked out there. I think that's a good estimate of, especially with limited resources, and these are $500 a sample. And most of the PFASs in the dissolved state, the only one that we found is bound to the particles was the PFOS. There's some, and then I could go off on the importance of how this relates to transport cycling and flowing down a stream and so forth, but there's a lot of ramifications. We could look at maybe differences in pore size. The reason why I chose that pore size was that was the TSS sample. Also, you could look at maybe if we put more mass on the filter, maybe we'd see some other compounds. But anyway, yeah. So there are probably seasonal differences. I do have seasonal data I haven't shown you. And also I have the surface microlayer, biota, and sediments not presented here to flesh this out more. So thanks.